I've been, I have been working with ILSI-IFBiC, the International Food Biotechnology Committee, for several years now. And the way they work is they identify problems or areas of concern that need to be addressed. They assemble task forces, and the task forces come up with a publication or some other way to address them. And then to complete the job, we're supposed to do outreach. We're supposed to go and explain what the task force has concluded and why they did it. And this activity is part of the outreach effort then. So the topic, the part on compositional analysis, I must give credit to Dr. Bill Price. He was at the FDA when the policy was first formulated. At that time, it was supposed to be for 5 years, and they were going to stop. But the people who implemented it retired, and the new people didn't remember. So we still do it. And then Angela Hendrickson-Culler from Monsanto, who made part of this presentation at a workshop we had in Argentina earlier this year and that Dr. Rao referred to. So before I start, some background information. I'm going to talk a little bit about crop variability, all the materials that Dr. Dapa and referred to. You know, how we domesticated plants, how much variability is out there, and how we use it for breeding engineering. Then how we use compositional analysis as part of the safety assessment. Then talk about some of the things that can interfere or affect the way compositional analysis is interpreted. And then provide a context for the role of the compositional assessment. So I want to start my story with actually with a summer vacation I got to do last year. I work on soybean, and they don't grow soybean in Europe, and I work on GMOs, and they don't like GMOs in Europe, so I very rarely get chances to go to Europe. And this was a vacation we took to Scotland, and went to this castle, which the Scots are very proud of it. They spent 10 million pounds restoring it exactly how it was in 1540, when a person called Mary of Guise lived there. Her daughter was very famous because she was Mary Stuart, and she had her head cut off in part of English history. But the point of the matter is that in this castle, they have reconstructed the lunch that Mary of Guise would have had in 1540. Does anybody notice something that should not be there? Yes, anybody? What's that? They actually had a good way to get fruits in from the Mediterranean and whatnot, but the thing that should not be there are the strawberries. At least not this type of strawberry. So I couldn't help myself, and I went to the fellow that's standing there, and I said, "You realize that they didn't have these strawberries in 1540? And he just looked at me very offended and said, "Well, here in Scotland, we have always had those strawberries." Of course, the people that I was with were just appalled that I would question it. But the point of the story is that most people think that the fruits and vegetables that we have today were found in nature in their current state. What this fellow told me is representative of the general point of view, and then it's no wonder that when we start engineering plants, it comes across as something new and different, rather than as part of an old process that we have been doing for many years. So these strawberries did not exist in 1540. In fact, the modern strawberries that we do have never existed in nature. We owe them to the French. We have to owe something to the French. In the area of global world exploration, they found a South American strawberry, which was kind of large, And the one from North America, which is really small, their fruits are about 1 centimeter in size. It was a cross of these two sometime in the 1740s, that's 200 years after the lunch in Scotland, that they came into being. And the same is true for a wide variety of crops. So nobody, many people will say, oh, somebody found a tomato walking in Italy and brought it to their garden, planted the seeds, and that's why the Italians use so many tomatoes in their cooking. Tomatoes are from South America, and what was found in the wild was a tiny little tomato. And it is then the process of going from that little tomato to the modern tomato that we plant breeders call genetic modification. And in the process, then, the past 50 years, breeders have been crossing it with other wild species and have brought about 50 different genes into tomato. This was an article that just came out as we were having the conference, the composition workshop in Argentina. And in that red, what they are is the circle represents the 12 chromosomes of tomato. And that red in the outer band is gene expression levels. In red is the gene expression level in cultivated tomato. And in black, you see the gene expression levels in ancestral tomato. And you can see the two of them pictured here. The point is that they're different. As we domesticated tomato, gene expression levels changed. And if there's differences in gene expression, that means that differences in composition have got to be present. That's just genetics at work. Going as far back as Darwin, Darwin actually wrote in his Origin of the Species that the crops that we have today are the product of selection and modification. And they're so modified that unless one is an expert in botany, they are most likely not able to recognize the wild versions of our crops. And that's what this picture shows. These are 3 wild versions of our crops. Anyone, let's get the agronomists not to answer, but for the rest of you, do you recognize them? Yes, no? What's that? Last one is the wild corn, Teosinte, yes. Okay, genetic modification. And the thing to keep in mind is that with the exception of some few changes due to epigenetics, it is impossible to change the appearance of something without changing the DNA underneath. And it is just now that we're starting to understand those changes at the DNA level that have taken place. So, the amount of genetic diversity present is amazing. If we look at SNPs, SNP is simply replacement of one nucleotide, like you can see there, or indels, insertions, deletions, where there's DNA present in one genotype and not in the other. We know now that in maize, the first time they just looked at 27 inbred lines, and just in those 27 genotypes, there were over 1.6 million SNPs and indels. And that was expanded a little bit more to include 103 lines, and now they have 55 million SNPs on the record. That's just SNPs, not counting indels. And yet, this wide variability in SNPs has never produced toxic or dangerous maize. So how much diversity is possible? This is, to put it in perspective, Ed Buckler at the University of Cornell came up with this slide. As different as we are in this room, we're not that different at the genetic level, but we're quite different from the nearest relatives, which are the chimpanzees. On the other hand, any 2 inbreds of maize are more different than people and chimpanzees. So the tolerance for DNA diversity is quite large. And that's what plant breeders have done, is this diversity has been the basis for plant breeding during the past century. And so conventional breeding is really about accumulating desired variation in simple varieties. One illustration is, they'll make 2 crosses, and the breeder over several years is going to be selecting anything that may not have the necessary disease resistances or growth habits is eliminated. Anything that lacks the necessary quality traits is eliminated. It better yield well over a wide area or it will be eliminated. And from those 1,000 crosses that they might make at the beginning, they'll end up with one variety that has an accumulated trait. Along the way, though, the vast majority are eliminated as undesirable traits. So there is this screening process they go through. However, the trait the breeder needs has got to be present. If the trait is not there, if they can, you mentioned, Dr. Rao alluded to the mutagenesis from the Atomic Agency. Sometimes they'll try to get the variability that way, but ultimately, if it's not, they have to find it somewhere else. So as early as the 1950s, breeders were resorting to gene transfer from non-sexually compatible species. As one example, in this case, they wanted to move one gene for rust resistance from this species here into wheat as the disease attacked both. The problem is that this species, Aegilops, would not cross with wheat. It will, however, cross with pasta wheat, which is a different species, it's a tetraploid. And once they had the hybrid there, they crossed the 2 types of wheat together. They made a back cross for many generations until they had recovered a plant that had all the chromosomes from wheat, plus 1 chromosome that came from the original Aegilops. And somewhere in this chromosome is that one gene they want. But in order to get the transfers, they resorted to X-rays. They broke the chromosomes up with X-rays and let the different pieces insert into the wheat genome at random. And the final product then is another variety of wheat. And if you notice, it does not look exactly like the parent. There's definitely differences there. And to this day, we know that the gene we wanted got transferred, but we have no clue what else got transferred. Obviously, a lot more got transferred because it looks different. Agronomically, it behaves different. And it's been apparent. It's been used in all the modern wheat breeding programs. So, but as I said, to this day, we don't know what is in it. But it's been safe. It's got a history of safe use. And similar techniques have been used in other crops. According to the literature, over 111 genes have been introduced in 19 crops just in the past 20 years. 55 in tomato, 12 each in rice and potato, and many dozens into wheat using similar technology. So that's sort of the background information. As Dr. Datta alluded to, though, we would prefer to move one gene and one gene only and not have all the other genes that might change the way a crop looks or behaves. And that's what gets us to recombinant DNA. I always use this cartoon to illustrate that it's very powerful technology. You can tell that by, you can do things that we couldn't do before. The cartoon illustrates that it's a technology that lends itself to exaggerations. As powerful as the technology is, we cannot create such level of changes. And if there's anyone in this room who thinks it's dangerous technology, there is your proof. The first victim of a GMO was Bugs Bunny. So once we start using RNA or recombinant DNA to introduce traits into crops, remember the perception was this is something new and different. The perception was we're taking plants from the wild and adding genes to them. And when we do that, there's two types of changes that we get. The first type is the intentional change. We want to add insect resistance, better oil quality, herbicide tolerance, whatever the trait is. And then that's fairly straightforward to assess the safety of these changes. Dr. Jess is going to talk about toxicity. Dr. Van Ree is going to talk about allergenicity. The Codex Alimentarius also added nutritional equivalents to the list of things to test for. But then there are the unintentional changes. One is that maybe the product we're adding can serve as a substrate to make a novel metabolite, or perhaps there's mutations that take place during the engineering process that we're just not aware of. So what is it that we are guarding against in terms of these mutations? This is a paper that appeared in Science back in 1992. And it claimed that plants have metabolic pathways that no longer function because of mutations that occurred during evolution. And these products, or the intermediates of them might be producing toxins. And these have been dormant since agriculture started, so we just don't know about them. But mutations that you make during the engineering process might activate these pathways by changing things, scrambling the DNA, by point mutations, those are SNPs, insertional mutations, the act of putting a transgene into the genome, or chromosomal rearrangements. So part of the assessment process is we avoid all SNPs. This was well before they knew that maize had 55 million SNPs. The question for today is, given that the first 55 million SNPs are not dangerous, why is the 55th million and first SNP a problem? We avoid filler DNA, or vector backbone sequences and whatnot. As long as fertility is not affected, there's no chromosomal rearrangements. And if we ensure there's no flanking genes nearby, then we are not going to be affecting expression of nearby genes. The problem is that we cannot exclude other mutations that happen elsewhere in the genome during the transformation process and which cause unintended effects. So there is no test for unknown differences. You have to infer the presence of unknown differences. And you infer these from differences in growth or differences in chemical composition based on a hypothesis. So that's what compositional analysis is all about, seeing if there's any differences in composition that might lead us to infer that other changes took place. And since then, compositional analysis is a key component of all safety assessments. Even when the regulatory systems are very different. Europe is process-based, very openly process-based. United States claims they're product-based, even though the trigger is still process-based. But as different as these two are, they both require composition data, where we look for increased or decreased levels of nutrients, anti-nutrients, things that we know about, and natural toxicants, plus composition in general that might indicate another change has taken place. The premise is as follows. Remember, we're looking for unintended changes that are also, or that could be hazardous. If we have two things that are identical to each other as the two halves of this potato, if they're identical, it is impossible for one to be safe and for the other one to be unsafe. But if we modify one, and then you say there's a risk associated with the modified one, that risk has to be coming from the difference. It cannot come from anything that was there before. So we start then by analyzing the composition to see what are the changes in composition that might cause a problem or indicate a problem is present. So the OECD has listed a lot of the key nutrients, anti-nutrients, and natural toxicants for a lot of the major crops. These are consensus documents. But for other crops, you probably need to get the information from the literature or come up with your own. Depending on the transgene and depending on the crop, the OECD documents might just be your starting point. If you're altering or the nutrition value or something else, you may need to add secondary metabolites to the list that you want to that you want to test. And as I mentioned, it depends on the trait. And everything should be driven by hypothesis. You're not going to test everything you can possibly test, but rather if you're altering a specific metabolic pathway, you would want to then target compounds in that metabolic pathway as opposed to unrelated things entirely. Whatever the composition data are, it is critical that they be replicated in time and space. They have the comparator. Usually the comparator is the isoline. Europe is now asking for the nearest non-GM relative. And you might even have some conventional varieties in there as a comparator. You need to grow them in the area where the crop will be grown. And they must be grown at the same time with the same practices to the extent that's possible. Harvest, storage, processing has to be in a manner that will maintain the integrity of the experimental material. You don't want it degrading before you get to the compositional analysis. Dr. Rao mentioned how essential it is to use validated analytical methods. And you got to use the correct statistics and interpret the statistics correctly. And I'll be getting to that point in a little bit. So if one looks at the list of compounds that OECD has for maize and soybean, it turns out that they only cover about 95% of the compounds present in soybean and about 90% of the compounds present in maize. And again, the list in the compounds in OECD were chosen to represent the products of key central metabolic pathways, with the premise that if there's a change in relative ratios, that could mean that an unintended change is present. The leftover fractions in both cases each consists of thousands and thousands of compounds, each one in very low quantities, so low that even if there might be a toxin in there, the amount is too low to be of significance. At the end of the day, dose makes the poison. For any of these compounds there to become a problem, we actually did the math. And for example, if something was as toxic as strychnine, it would have to be about 5% of dry weight before it would have a toxicological effect on whatever was eating it. And for it to be present in such amount, you would certainly detect the alterations during the compositional analysis that is done. We like to think that if we have two plants, GM and the isoline of the GM, the comparator, that you can plant the two of them side by side and that you're going to get the same values. But that rarely happens because there's a lot of compositional variability, which can come from genetics, the way things are planted, and even the environment themselves. So when you're going to compare two historical values, these have got to be taken into consideration. And a lot of the material that I'm going to present came from the ILSI-IFBiC workshop on composition that we had in September of last year in Washington. Things vary. What you can see in this table is that you have one variety of soybean planted the same year in 4 different cities, and isoflavones, which are physiologically active in mammals, vary, there's a 4-fold variation in their level. Alternatively, one can take 4 varieties of soybean, same place, same time, and you get a 3-fold level in the amount of isoflavones. So that's sort of the background level of variability that we're talking about. This was just for any of these species down here. These are just different, the natural variability that exists for folate. And you can sort of name your metabolite, and there will be that range of variability. Just segregation that happens during a breeding program. This is just from crossing 2 different inbreds together and looking at the progeny. In the progeny, there's a 2-fold amount in the amount of iron present. These are dry beans. And likewise in the range of zinc. So this would be an example of genetics segregating out a trait. We've been changing things over time as well. A phenomenon that's happening in corn or in maize, and it's also happening in soybean, is that we're harvesting less protein per seed. We're harvesting more protein per hectare, but because we're getting more seeds per hectare, so we harvest more protein per hectare, but it's decreasing per seed. So, this is a problem. If you have to use, a brand new hybrid that's a GM, might be over here. But if we're starting to have to use non-GM comparators that might represent genetics from this era. This is just the levels of tocopherol, that's vitamin E, decreasing as more phosphorus fertilizer was added to the field. And that's not the only thing. The number of seeds per hectare and the row spacing also affects something like tocopherol, and this is all within one variety, keeping variety constant. This one's in wheat, data from Peter Shuri. 26 varieties of wheat, and when you cluster them, they cluster by where they're grown. So those 26 varieties, are, I'm not sure how to go back on this. Yeah. 26 varieties cluster according to where they were grown. You can see the different colors. They cluster by where they were grown, not by how related they are to each other. So that's the effect then the environment has on composition. Even the heterogeneity within a field. Just looking at a field, this is a 20-hectare field that has changes in elevation. It can change protein from less than 34 to 38. This would be a significant difference in a compositional analysis. And 18 to and a couple percentages just in the oil in a small area. So we have to interpret those results. So if one has done the compositional analysis, and the first question for each individual component is it statistically different from the comparator? If it's not, we say it's as safe as the conventional. But if it is different, and just remember, when we do statistics, we use the 5% significance level. That means that by chance alone, 5% of our comparisons should be significant. In that case, we look at how much variability is present in the comparator. And conventional crop varieties that have a history of safe use, values reported in the literature, in the databases that are available from different sources, with the ILSI database being by far the largest and most comprehensive of databases. So if the variability, if the new value is within the range of one of the above, we go back and can say it's as safe as conventional. But if it's not, we first have to ask the question if that difference is biologically relevant. And if it is a biologically relevant difference, it has to be, a valid explanation has got to be given, or you go back and generate new safety data, and if that doesn't work, that event is not going to be approved. So to use variability to provide a context for compositional assessments, we're going to sort of talk a little bit about the difference between a statistical significance and a biological relevance. And that's done by using variability to provide the context. In this particular example of some isoflavones in soybean, we compare the levels in the comparator and in the GM, and there will be one in this case that statistically is different in terms of content. But the question is this a relevant difference? Tukey, the great statistician, always mentioned that for any time you do a comparison, depending on how many decimal places you want to get to, there's always going to be a different. So we have to consider what the end goal is, and the design of the experiment, how big it was, and how variable the data are. Here's an example that's being used in schools now to illustrate the difference between biological relevance and statistical difference. In the United States and in Europe, when the little children ask their parents, where do babies come from? They say the stork, this bird here, is the one that delivers the babies. Any lady that's had to carry a baby before knows this is not true, but it's a great explanation for kids. Well, what do they tell children in India when they ask where children come from? What's this? Same thing? Okay. Well, in this case, what they did is they looked at the number of storks and the birth rate in different countries in Europe, and they did a regression here and come out with a p-value of 0.008, highly significant. So what it really means is there is a 1 chance in 125 that the results are due to random chance alone. But a lot of people, though, misinterpret this, and the misinterpretation is that there is 124 out of 125 chances. That's a 99.2% chance that the storks deliver the babies. Okay? So keep that one in mind as we distinguish. Just because something is relevant, this doesn't mean something is statistically significant does not automatically mean, it can mean, but not automatic, biological relevance. So the statistical analysis is what identifies the differences for us. In this example, you have the mean difference in the transgenic and the control of about 7 micrograms per dry gram, per gram of dry weight. If we compare this difference to the amount that's naturally present in the varieties, it's well within that range. So looking at the magnitude of the variabilities is what provides a context for biological relevance. So you still have to come back. Where do you find out what's the natural variability? And that's where the databases come in. As I mentioned, the ILSI database, it's got over 100,000 data points on 3 main crops. They're in the process of adding rice to it, and they're adding canola to it, and I think potato are being added to it right now. And they're adding data points from different parts of the world as well. It's got user interface. It has improved considerably this past few years, and it's free public access, so it's always there. So the basic premise is that to get the context of whether something is biologically relevant or not, we look at the differences. So we get back to the difference in the transgenic and the comparator, the amount within the comparator itself, the ones in the literature and the ones in the ILSI database. So this tells us that we're well within the normal range. So the key thing too is when everything here comes from a variety somewhere, and it represents levels of the particular analyte that have a history of safe use. Someone around the world has been consuming those. Bill Price went and he reviewed 129 dossiers from 11 different crops that were submitted to the FDA with compositional data in them. And FDA ruled that all 129 were not materially different from composition, safety, or any other relevant factor of varieties now grown, marketed, or consumed in the US. In other words, they were substantially equivalent. And for the record, no other country that's looked at compositional data and looked at variability has found differences either. Why is it that we're not finding these differences, given the diversity of GM crops out there? A variety of reasons. One is that there are many steps in the process where any unintended effect is going to be eliminated. I'm going to do this, but during the transgenic selection, when these are back crossed into different varieties, and even during seed production. To show you in more detail, I mentioned during breeding that a breeder is going to cross two plants, get thousands of F1s, get hundreds of thousands of F2 seeds, narrow them down to one. In transgenics, nobody makes one transgenic event in the laboratory and says this is what we're going to sell. They're going to make hundreds to thousands of transgenics. Anything that does not express the gene or is going to be eliminated. Anything that has an off phenotype is going to be thrown out. If the gene's not there stably expressed over 4 generations, it goes out. And then these get nailed down to just a few that are potential lead events. That's that they're going to be commercialized. And they start looking at composition, agronomic, and all that gets narrowed down to just one. So this is one area where a lot of possible unintended changes are eliminated. Secondly, during the breeding process, you take a transgenic, cross it to a non-transgenic, the back cross, and the amount of red, in this case, that's the amount of traits coming from the engineered plant, where possibly the unintended effect could have happened, it gets eliminated by half with each generation until by the time you have something commercial, there's very little of the original genome left except for a few bits of DNA around the transgene. Seed production, all Western countries, the breeder has a small bit of seed, and that's planted under very controlled conditions, and then that's harvested and planted again to produce certified seed that is sold to the farmers. And at every stage of the way, anytime a plant looks off, it's thrown out. So one is the process that is built to eliminate unintended changes, whether they come from conventional or GM breeding. Another one, and I think probably the major reason, is that conventional breeding, genetic modification per se, does not have that big ability to produce unknown metabolic or unknown toxic substances. One of the things we did with the task force is we went through the literature, spent a lot of time reviewing all the breeding literature, finding events where conventional breeding led to problems. And of the millions of conventional varieties out there, only 9 have been reported to have unintended effects, and that they gave either a skin rash or stomach aches. No one ever died from a cultivar that somebody developed. So that makes it one of the safest human technologies. Of the ones that gave problems, all of them involved elevated levels of known toxins. Things that were on or are on the OECD list. So as part of the breeding process, anytime anybody works with a crop that has known toxins in it, and it doesn't matter if one is doing transgenics or conventional, known toxins are monitored to make sure that we don't inadvertently raise them. But again, the concern here for the compositional analysis is the unknowns. And in all the history of breeding, we were not able to verify a single case where a brand new toxin that did not exist at the genus level appeared spontaneously. Not one. But in the interest of full disclosure, the EU documents do cite one example. They cite the case of Lorilla et al. In 1996, where they found a novel toxin in a potato somatic hybrid, and they are using that as proof that novel toxins can appear. The only problem is that that very same toxin was described in potato in 1981. So I get back to my premise. In all of breeding, we have never come up with an unknown toxin. So when one compares the techniques of conventional breeding, mutagenesis, and transgenic, there's a technique called comparative genome hybridization, whereby one takes a genome from a plant and labels it with a fluorochrome, say a blue dye, and if one has mutagenized the plant, the plant that has been mutagenized is that genome is hybridized to a dye with a different color. And then if you mix the two of them together, when the DNA binds, the two colors cancel each other out, unless there's a piece missing in one genome. And if there's a piece missing in one genome, then we get a signal because one fluorochrome cannot be canceled out by the other. So this is a case of two soybean plants, the parent and one that was subjected to fast neutron radiation, and indeed there's 6,000 base pairs that are missing. Compare that to two varieties. Huge levels of indels naturally present. And compare that to transgenesis. We can't detect anything above background noise. So it gets back to the breeding really does make far more changes than we can possibly do with transgenics. So to start summing up, how does variability impact our safety assessments of GM crops? Keep in mind, we consume variability every day. You know, nobody minds eating a dragon fruit in the morning and rambutan later, even though compositionally they're not alike at all. But we tend to concern if the lemon has a little bit more citric acid than the lemon we ate yesterday. So there is a conceptual issue here that some of the communications efforts need to work on. So we do start by defining variability. It shows what we have safely as human beings been consuming for a long time. And then evaluating the relevance of variability from GM involves considering it in the context of natural variation. Natural variation really meaning what has been safely consumed, what has a history of safe use. So the take-home messages are that compositional analysis is there as an additional safeguard against unintended changes, above and beyond what's done with the toxicology, allergenicity, nutritional equivalence, and whatnot. But the probability of an unwanted change to begin with is pretty low. Crops have been extensively modified during domestication, and Dr. Datta mentioned that, and subsequent breeding. And pretty much the vast majority of changes in composition have not been dangerous unless we elevate known toxins. In general, novel metabolic pathways are not going to materialize out of thin air. The genes have got to be there and expressed. So really, breeding is not known for the generation of novel toxins. And the system is in place that eliminates unwanted types at many steps of the process. Engineering makes fewer changes than breeding does. And at the end of the day, genotype and environment are going to contribute far more to compositional variability than an insertion of a transgene ever will. So with that, this is the task force on compositional analysis that IFBiC put together. All IFBiC task forces have to be tripartite, members from academia, from government, and industry. And the entire conference was videotaped, and all the videos are online, and you can go to this link and download them. You can download everybody's presentation from it. We had speakers from all over the world. And then the speakers were asked to submit a manuscript, and all the manuscripts then have been published 2 weeks ago in the Journal of Agricultural and Food Chemistry and Plant Physiology. That's where the volume and issue and page numbers and all the PDFs are going to be available from the ILSI-IFBiC website. So with that, thanks so much for the attention.